So we're going to understand, drug action, right? Drugs are going to interact with proteins, right? And that includes, again, receptors and also transporters, ion channels, and enzymes, right? Many of these proteins that drugs are going to interact with are located on the surface of cells, right? They actually are embedded in the skin of the cell or the cell membrane. So it's important to understand a little bit about the structure of that cell membrane. And what's remarkable about cell membranes is that regardless of where in the body we're discussing, the tip of your toe, the heart cell, a neuron, these membranes are made of the same stuff that has very specific sort of functional properties, okay? So structurally, it's the same, and it has a particular function that it'll share regardless of which cell we're discussing. So this is a little chunk, little piece, a cross-section through, right, the membrane of a cell, of any cell. And you'll notice it's all these little ball and stick figures. There's actually two layers, right, of these sort of ball and stick figures that are represented here. Each one of these little ball and sticks is referred to as a phospholipid. That's the type of chemical that it is. And it's a very unusual and very interesting chemical in that it has a charged, head. It's made out of phosphate, right? So it's got a negative charge on that, ball portion of this ball and stick molecule. And then it has a long sort of lipid tail. This is an uncharged kind of fatty tail. Lipid is a fat, right? And I want you to think about if you were to take something that's charged, let's say water, right? H2O has a positive hydrogen end and a negative oxygen end, and you were to add to it oil, which is fat, right? Which is lipid, which is uncharged. There'll be a very clear separation between the water layer and the oil layer, the charged and the uncharged. And this is exactly the behavior that you see exhibited by these phospholipids. If you throw them into a typical bodily environment, which is largely charged, mostly water, right? These will line up, in two layers, what we refer to as a bilayer. This allows for, let's say, the inside of the cell, the phosphate heads that are charged to be oriented inside, right, with other charged constituents in here, okay? And the phosphate heads in this layer are, oriented out to the extracellular space, outside the cell, right, to the other charged constituents out here, mostly water, right? But the lipid fatty tails that are uncharged form a layer. They're isolated in the center of that membrane. And this is a very, kind of stable structure. And it also has the property of being impermeable, which means it doesn't allow the passage of charged substances from one side to the other. From either the extracellular to the intracellular or the intracellular to the extracellular space. It blocks things from going back and forth, okay? Now, the way things get through this impermeable phospholipid bilayer cell membrane, right, is via these specific protein structures. Remember, these are manufactured based upon instructions in your genetic material, and they are embedded in the membrane here. And these protein machines, which again include receptors, channels, transporters, and a pump, okay, I'll explain what that means a little bit. That's another kind of machine, are embedded in this membrane and allow for the passage of certain substances across it.